I also think that I am the last person before your break, and I am keenly aware of that. So here I am with my phone. I'm going to set my timer here. No one likes that person. All right. So I am going to talk just a little bit about some of the really cool plasma-based biomarker work that we are doing here at COMBINEDBrain. And it includes, obviously, kids with SLC6A1 related disorder. Okay, so I have a couple disclosures, but mainly that this project was funded by 7 different industry partners that are all investing in the rare disease space, including SLC6A1 related disorder, which is a really exciting time for me. Okay, here's the proverbial slide that I show every time I present, and it really just is a nice schematic of an overview of the drug development process. And I'm not going to go into great detail on each of these steps. There's no pointer here, but is there a pointer? Oh, there is a pointer. Okay, in the middle of this part, one critical part of the drug development process is the identification of biomarkers, right? And I know that I don't need to go into a lot of detail about what biomarkers are in this group, but so I will go over this pretty quickly. But basically, biomarkers are objective measures that you can use to predict disease status, progression, and or how well an individual will respond to a particular treatment. And these can be mRNAs, these can be epigenetic signatures, these can be proteins, metabolites, microbiota, or these can be physiologic markers, so electroencephalograms, like somebody I think have already presented or will present later. So like I said, most of the time, or quite often, these are biological fluids of some variety, like blood, urine, cerebrospinal fluid, or again, physiologic EEGs, ERPs, or even nerve biopsies. And for this project, we really focus on blood-based proteins as potential biomarkers for some of these rare genetic neurodevelopmental disorders, including SLC6A1 related disorder. So the main aims for this study were to identify neurodevelopmental disorders with shared plasma proteomic profile, as well as unique proteomic profiles by disorder. The second aim was to understand the impact of age and epilepsy on plasma proteins, as well as to understand the impact of specific SLC6A1 genetic variants, so missense variants, nonsense variants, etc. And then the last thing is really to start honing in on some of these proteins to see which ones correlate better with indicators of disease severity, many of which have already been talked about in some of the previous talks. So actually I'm going to go ahead one slide. Okay, there's my picture. So the study design for how we were to approach this is fairly simple. Our goal was to try and collect 20 blood samples from each neurodevelopmental disorder, as well as 40 unaffected, relatively aged matched controls. In addition to getting their genetic report, so we could look at their genetic variant and confirm specific diagnoses, as well as supplemental genetic information, we also had them fill out a number of neurodevelopmental surveys, everybody's favorite caregiver reported Vineland, as well as the Pell's seizure survey developed by Dr. Greenspan in the room. And then we got a couple of other pieces about genetic medical history as well. And this is just a schematic of how we did this. So we recruited mainly through the PEG, so people like Amber we would harass incessantly to try and help us get 20 people with SLC6A1 related disorder. Some of our wonderful biorepository coordinators would consent those participants. Again, we would collect genetic reports and have them fill out neurodevelopmental assessments. We would collect a couple different vials of blood and then ultimately process them as plasma. In addition to some of the developmental assessments, we have a variety of what I call day of clinical assessments. When's the last time they ate? When's the last time they had a seizure? If they have seizures, are they on any special diet, etc. Things that you would think might affect the proteins that are circulating in a kid's plasma. Okay, so this is how we collected those samples. So the vast majority of how we collected these samples is we flew to different family conferences. And so you can see this schedule right here. I like to refer to this as a very less cool Taylor Swift Eras tour. Basically around the same number of locations, less sold out, but just as successful. So we would fly a team to these sites at these conferences, and people would come in, collect blood from their kid, and we would process it immediately and freeze it, and then we would use a small portion of that for the proteomics study that I'm about to show you. For kids who are more severe or couldn't otherwise come to these conferences, we had phlebotomists come to their home. And then the last thing that we did was we worked with a lot of clinicians who were fantastic. Two of the ones that really helped us was Jackie Harris and Liz Berry Kravitz, almost forgot her name on the spot, who really helped us collect blood in tandem with clinical labs or other research projects that they were doing at the time at their own institution. Okay, so this is kind of a hefty table, but what this is the total number of patients or samples that we recruited for this study, and it's about 700, a little bit over 700 total. I'm not sure how closely this font shows up, but you can see this little purple-pink box here I put over. The number of patients we had for 6601 related disorder was 15. The average age was 10. Percent female in this cohort was 27. And then we also asked a variety of different questions about their phenotype. 11 of these kids, or 82% of these kids, had seizures. And here is another way to show total enrollment. Again, enrollment for 6601 related disorder is right here in this pinkish bar right here. The bluish purple are the affected individuals, and then we also tried to collect from unaffected siblings to try and get a well-matched, age-matched control. But as you can imagine, it is sometimes significantly more difficult to collect a blood sample from a kid who doesn't normally have blood samples. So in this case, for any data that I show that looks at comparisons between 6601 related disorder kids and controls, those are unrelated but age matched control samples. Okay, so one of the first things that I think about when I think about looking for biomarkers for a particular disorder, and in this case, a biomarker, potential shared biomarkers across 30 different neurodevelopmental disorders, is how does that protein change with age? And even more importantly, how does that protein change with age differently between kids who are affected versus kids who are not affected? And so that's what this volcano plot starts looking at right here. As you can see, the dots sort of on the upper end of this figure right here. Let me see if I can just move, it'll make more sense. The sort of less blue colored dots right here, those are significantly differentially expressed proteins with age in all of those affected populations that we were looking at in this study. And I've circled just a couple of them here at the top. This is WISP2, which is a Wnt signaling related protein, and this OMD, which is an osteomodulin, is involved in bone formation. And when you look at the graphs over here on the right-hand side, which are just mapping those two proteins that we found that are significantly changed with age, you can see, in fact, that for this protein, WISP2, it significantly increases expression during development and then sort of tapers off into adulthood. And then you see this opposite trend with osteomodulin, which is the bone formation protein, during development, and then it sort of peters off in adulthood as well. So the next thing that I wanted to look at was, okay, well, what proteins change differently in affected kids versus controls with age? So here's the same figure layout. Again, the individual marks here are proteins. Here, I remembered to put a line that showed me the significance. So any dots are proteins that are significantly changed with age differently between affected individuals versus controls. And I've labeled a couple of the ones here at the top. Again, some of the most greatly changed are the cyclic AMP-dependent transcription factor, ATF6 alpha, this complement-related factor, and then adiponectin. So these two on the right-hand side are upregulated in affected kids, and ATF6 alpha is downregulated in affected kids. You do not need to read this small print right here. This is just merely a figure of showing you the extent to which those proteins are upregulated or downregulated in individual disorders. So these are the individual disorders written down here in very, very small font. And then the pink arrow right here is SLC6A1 related disorder. So you can see, in fact, adiponectin is up in kids compared to healthy controls. So is a complement-related factor, and then ATF6 is technically lower in SLC6A1 related disorder kids compared to controls. Okay, so the next thing that I was thinking as well, there are some shared pathways and shared proteins, but do these overall, does the plasma proteomic profile by different disorders, are they different from each other? So do the plasma proteins in SLC6A1 related disorder change differently from SYNGAP kids or kids with MED13L or CTNNB1? On what you can see here, this is a principal components analysis. All of these different colors are the different disorders in this project. And you can see that there's no real clear differentiation amongst disorders. The blue dots, the green dots, the red dots, the orange dots are all really fairly evenly distributed amongst each other. So there's no clear differences in plasma proteome amongst the neurodevelopmental disorders that we were looking at. And just to look at this a little more fine-tuned for specifically for SLC6A1 related disorder samples. So here, the black dots are the control samples here that still sort of have this Nike swoosh shape. This is a really terrible color scheme here, but the brown dots, which no one in their right mind is probably going to be able to see as I realize this, these brown dots show a fairly similar trend as the control samples as well. And then, because I didn't think about this ahead of time, I changed the color scheme again. And here you see the black dots are the control samples. Now, the red dots right here are kids with SLC6A1 that have epilepsy, and the green dots are the kids who don't have epilepsy. And you can see that most of the red dots sort of cluster a little bit over here, whereas the red, or what color is that? Green. The green individuals without epilepsy look like they may start distinguishing from those kids who do have epilepsy. Okay, so sort of trying to take a closer peek at what proteins are specifically changed between kids who have SLC6A1 compared to age-matched controls. Here is a similar volcano plot to what I showed before, but in this case I'm just looking at SLC6A1 kids compared to age-matched controls. And again, the same sort of format is the same. Fold change on the x-axis, and on the y-axis is our false discovery rate. The pinkish purple dots right here represent proteins that are significantly differentially altered in kids with SLC6A1 compared to controls. In total, there are about 108 differentially expressed proteins. 46 of those are downregulated, and 62 of them are upregulated. I labeled a couple of them here: MORN3, IGF-1, VEGF, PYY, GIP. those are some of the ones that I just pulled out as I noticed and recognized as being significantly differentially expressed. Again, the ones that are on the left-hand side of this volcano plot are downregulated, and the ones on the right-hand side of this volcano plot are upregulated in kids with 6601. So just to look at this in a different way, I pulled a handful of those significant plasma proteins out, and you can see that plasma VEGF, GIP, and PYY are significantly, albeit modestly, upregulated in plasma of 6601 kids. Conversely, plasma IGF is actually downregulated in kids with 6601. So, okay. So I actually did one more thing, which is to start looking at, so IGF I got a little bit excited about, mainly because I recognize that. So the last thing I wanted to look at was, I wonder how IGF changes with age, which is one of the first things that I started looking at in this project. Whoops. Aw, boo. Oh, wait a minute. Huh, those are not the slides. Okay, bummer. Okay, so I'll wing it. When you look at the slides for 6601 and IGF levels, what you see is that IGF goes down with age in kids, whereas IGF stays the same with age in the age-matched control population that we look at. Which is really interesting to suggest that IGF-1, amongst the other proteins that came out from this project, might be something that would be specific to look at and measure in 6601 kids compared to controls. Okay, so we identified 108 plasma proteins that are altered in 6601-related disorder compared to controls. And based upon those top proteins that we looked at, it looked like it would be involved in gut peptide, hormone regulation, insulin signaling, as well as perhaps cilia function. So the next steps for this are really to look at the impact of genetic variant, focusing in on differences in kids with missense versus nonsense versus intronic variants in plasma proteins, to identify proteins that correlate with indicators of disease severity, such as Vineland and the seizure survey or seizure burden. And then lastly is to collect longitudinal samples, which is what we're doing here today, to start looking at how these proteins change over time. Okay, so I would say this is our team at COMBINEDBrain here. I would say just about everybody has had their hand in helping with this project. The people that I should really be thanking are obviously not in the room because they're over there actually collecting samples right now. But so if you guys see them, just say that I acknowledged them appropriately and gave them credit. And that would be people like Katie Schmidt, Rachel Heilman, Sasha. I'm gonna forget some people. Let's just say everybody has been incredibly helpful, as well as our fearless leader, Terry Jo. Obviously, I couldn't have gotten any of these samples without all of the patient advocacy groups being fantastic and helping us recruit and get people to our roadshow collection sites, and then obviously our fantastic industry partners who are just wonderful to work with and are really encouraging about their excitement for working with rare diseases. So I'm very grateful to them. And I don't actually think that I have 30 seconds. So there you go. All right.